Cancer Epidemiology seminar, and for those of you who have not been at the previous ones, this is our event each year where we pay special tribute to Bob and his many contributions to cancer epidemiology, and especially his one of the main areas of focus, breast cancer epidemiology. I think I can speak for many of us that Bob is still really greatly missed. Again, for those of you who didn't know Bob, he's an exceptional intellectual force, an exceptional mentor, exceptional inspiring teacher. He made just so many other contributions beyond just his role in the department to the school. He worked very closely with a number of breast cancer advocacy groups, and I can say confidence that he touched the lives of many students, colleagues, and friends. And so through this seminar, we're committed to honoring Bob's memory. We're also, for those who work on CBCS, are committed to ensuring that his study, the Carolina Breast Cancer Study, still is as great and as influential as ever. We also remember Bob, I'm sure each of us in various small ways, recounting amusing things that Bob did and other events that Bob was involved in at work and outside of work. And this year's event is even more special than usual, that we're very pleased to announce the first awarding of the Robert C. Millikan Cancer Epidemiology Award, and this is awarded to an outstanding student, graduate student in our department who's working on and has a strong commitment to breast cancer epidemiology. And so, yeah, I'm pleased to announce this year, 2017, winner of this award is Nikki Niehoff. Nikki really has been, this was a no-brainer. Nikki is an outstanding student in the classroom, is fully committed to cancer epidemiology research, has already published a number of papers, cancer epidemiology, including breast cancer epi studies, and really is truly most deserving of this award. This award is made possible by a number of generous donations made by a variety of folks, but want to especially note that Bob's family has made a very generous donation to the department to sustain this award for many years to come. And I will ask Bob's sister Ann to wave or stand up, and let's give her an award. Thank you for coming, and again, thank you so much to you and your family for this great generous donation to the department. So, as is the tradition here, make Nikki take the long walk to get her certificate and a check that I wouldn't try and cash this check quite yet. So it's a pleasure to introduce today's Millikan Seminar speaker, and we're very grateful that he flew across the country to give this seminar and spend a lot of time today with various training grants and students and colleagues in the department. And that's Dr. Joan Witte. John has multiple roles. I have a tough time keeping track of all your roles here at University of California, San Francisco. He's professor of epidemiology and biostatistics, also professor of urology. He's co-leader of the program in cancer genetics at the Helen Diller Cancer Center. He is also associate director for the Institute for Human Genetics and head of the Division of Genetic and Cancer Epidemiology. And for those who don't know John's work, he's really a national, international leader in the synthesis of methodologic and applied research in genetic epidemiology. His current work in his lab is really on the underpinnings of methods for analyzing next-gen sequencing data and GWAS data, and integrating with other forms of omics and other data. And I think he'll talk about some of his areas of focus today, but it's been particularly on prostate cancer, pharmacogenetics, and birth defects. He has too many awards and other things to name, so I will just turn it over now to John. Thank you so much for coming.
Thanks, Annie, for the nice introduction, and also for the invitation to come and speak. So I'm going to be talking about the genetic epidemiology of prostate-specific antigen screening. And so this is actually an area that I work a lot in prostate cancer, and PSA is a tool that's been used quite a bit to screen men to see if they have cancer. Fairly controversial screening tool, and what I'm going to be talking about today is whether or not we can incorporate genetic information to try to improve the sensitivity and specificity of the PSA measurement. So PSA, it's a glycoprotein produced by the prostate, and it actually generally, so this little diagram on the right shows the prostate ducts. Usually PSA is released into the ducts, but sometimes when there's something going on with the prostate, generally, say you have cancer or you have hyperplasia or prostatitis, the PSA can actually also go into the bloodstream. Right, so it's going to be picked up with a blood test, and is quite good at monitoring cancer. So if you already have cancer and they want to see whether or not it's progressing or is a recurrence, using PSA is a very good measure for that. What it's less good at is trying to determine whether or not someone actually has cancer. And that's in part because it can be increased due to other factors. And this has led to a lot of kind of discussion about whether or not it should be used, and whether or not the use of PSA has led to overdiagnosis of prostate cancer and overtreatment. Now, it's a little bit strange to think of overdiagnosing someone for cancer, but with prostate cancer, as men get older, they're more and more likely to have some latent prostate cancer that may not actually cause any morbidity or mortality for them. But once you go in and start, trying to detect cancer, you find the cancer, then these men are often treated, and there are a lot of side effects, and they may or may not have needed to be treated. So I'm going to walk through kind of what happens once a man is PSA screened. So you've got these different decision points that you can make at times. So first of all, whether or not you're going to have PSA screening, no or yes. And if you do, if you have an elevated PSA. And if you have an elevated PSA, should you have a biopsy for prostate cancer? And if you have a biopsy, do you have cancer? If you have cancer, whether or not you should be treated. And I say this, one of the treatments now for prostate cancer is called active surveillance, where you actually don't get treated. You keep an eye on men to make sure that they don't progress. And really, as I mentioned, what I'm going to talk about today is the genetics. Can we incorporate genetic information to these early steps in this kind of cascading series of events? People often talk about PSA screening as just getting you into this medical system that just progresses forward. And once people start looking for cancer in the prostate, they look hard enough and you're old enough, they may very well find it. That said, a lot of men die from prostate cancer as well. And so finding it early is important. So there was 2 clinical trials that investigated whether or not screening PSA has a benefit with regard to not cancer, but this endpoint here, and that is death due to prostate cancer. And they had very different results with regard to mortality benefit. So there was a European trial and a US trial. Can anyone tell me why they saw such different results? Anyone who's familiar with this? The European trial actually did see a mortality benefit due to PSA screening, where the PLCO trial did not.
I think an inadvertent crossover in the control arm in the USA
right.
trial.
Yep. So the men, because they were in the US, and because PSA screening is for a lot of men, is just an annual thing that's done, a lot of the men who were in the non-screening arm got screened. right? So I mean, a lot, a huge, something like half or more. So it ended up having this effect where it washed out any effect at all on mortality. And what this led to was a grade by the U.S. Preventive Services Task Force of a D. Now, a D here is like a D in your classes. It's not good. And in fact, it means that the harms of screening outweigh the benefits, which means screening is no longer going to be paid for by insurance companies. They're saying, this is bad, we're not going to screen. What this led to then was a decrease in screening. You can actually see that it's been 5 years or so now. You can see that PSA screening has really decreased. The incidence of prostate cancer has gone down too. Imagine that. But you know what else has gone up? Deaths due to prostate cancer have actually, we can see it in the data, they have increased. And so the European trial has continued to reanalyze their data and publish papers, and they've shown that this mortality benefit is getting larger and larger over time, which suggests that the screening actually is good for mortality. And there's actually a paper that just came out in September here that did a nice analysis. They did kind of a counterfactual analysis where they're looking at the lead time for men who were clinically, trying to get what's the difference between if you just had clinical diagnosis versus screen-detected diagnosis. So what's the difference between those two? And they were actually able to analyze the data from the two different trials and showed, lo and behold, that screening is actually a benefit for mortality due to prostate cancer. So what did that lead to? Well, the task force met again just recently, and they upgraded it to a C. Now this hasn't been finalized. Yeah, so like school, you can bump back up, so they went up to a C. And it's still this is under discussion. So if you go look online for the task force, it will come up as a D, but then there's a thing that says, this is under discussion. Click here to see the C. And what they're recommending is that men between the ages of 55 and 70 should, what does it say here? Clinicians should inform them about the potential benefits and harms of PSA screening for prostate cancer. So kind of a more, let's not just not screen at all, but let's have a discussion with your physician about the pros and cons and make a decision for the screening. So you've got this now age group, and in fact, the way screening works in general for prostate cancer is they have different cutoffs to decide whether or not to move on to a biopsy depending on your age. Okay? Because as you get older, your PSA increases. So it's not a one-size-fits-all. And not only that, they also want to know about your family history of prostate cancer and your ethnicity when they make this decision about how to interpret, whether it's a PSA screen and how to interpret the results. Well, once you start talking about family history and ethnicity, that starts making you think, well, wait a minute, what about just genetics? Okay? Now, people are different, and men have different PSA levels based on just their genetics. So there's been some studies looking at the heritability of PSA levels to show that it's a fairly highly heritable. So the narrow sense heritability here is, it's been estimated to be about 40–45%, which is quite high, actually. And so what I'm going to explore here, as I mentioned, is whether or not we can actually use genetic information to kind of normalize men's PSA levels to decide whether or not they should go on and have a biopsy. So it may very well be that I'm just a high PSA producer. So my threshold to decide the next step maybe should be higher than someone who's a low PSA producer. So there's been a couple of Genome-Wide Association studies of PSA levels. One, the Icelandic group Decode did a big Genome-Wide Association study, and they were actually interested in specifically this question, and that is the clinical translational value of knowing about the genetic information. And they came up with 4 single nucleotide polymorphisms that they felt could actually, that were associated with PSA levels. And then there's been a small Genome-Wide Association study of Asians. And what I did actually was to move into a much bigger study. Both of these studies were fairly small, so you're detecting like 1 or 2 genetic variants. Now these genetic variants have very weak effects, right? So the odds ratios are like 1.1, something like that. Well, here we're talking about a continuous variable, but they're very small increases in PSA levels that they predict. So what you need to actually explain more of this genetic basis is big studies, large studies. So I collaborated with people at Kaiser Permanente in Northern California. Now they have a cohort of 100,000 people that they have genotyped using an Affymetrix array, and about 40% of those individuals are men. There are some men with prostate cancer, but there's a lot of men without prostate cancer, and because they're a large health organization, they have PSA measures. A lot of PSA measures, like 500,000 PSA measures on 30,000 men without cancer. And so what we were able to do is actually go and pull from their medical records all of their PSA, so their complete PSA history, even on men with prostate cancer. What was their PSA history before they were diagnosed? And there's 4 different things that I'll talk about here. First is just the Genome-Wide Association study of PSA levels that we undertook with the Kaiser data. Then trying to differentiate between PSA genes and prostate cancer genes. Because there's a bit of an issue here that if you get screened for cancer because you have high PSA, that those genes are going to show up. PSA genes are also going to show up as prostate cancer genes, because that's how you get the decision to be screened. And then the what we call the chip heritability of PSA, to compare it to those previous estimates of heritability. Now, this is something you could do with Genome-Wide Association study data. And then finally, what happens if we try to do this normalization of PSA levels? How are men with and without cancer reclassified? And how do they move around in this kind of decision area of whether or not they should get biopsy? Feel free at any time to stop and interrupt or ask questions. Okay, so Genome-Wide Association study. The idea here with these studies is that you measure, you take a little array, a chip, you measure it on everyone and get hundreds of thousands, millions of genetic variants, mostly common genetic variants across the human genome, and you associate those with the trait. In this case, we're interested in PSA levels, as that's our outcome. And we're just trying to basically, very simple analysis. In this case, we actually did a GEE model because we had multiple measures for our dependent variable. We had multiple different PSA measures. And so we weren't actually looking at the slope change over time. We were more, it effectively is kind of like looking at the median level of PSA. Now I should say, PSA levels can go up, say if you have prostatitis, and come back down. And you never have cancer at all. And so this study we had, we focused first on the non-Hispanic whites in our study, what we call our discovery. So that's what we're trying to detect our initial associations between these genetic variants and PSA levels. And then, of course, with these types of studies, you need to replicate them. And so we had a number of different populations. We used all of the other ethnic populations within Kaiser. And also, we actually used the European ancestry cases and used their earliest pre-diagnostic PSA level. And then we had 3 external control groups that we also looked at to replicate these results. And so this is just a little descriptive table showing you what their ages were within these different populations and what their PSAs were. And you can see there's one difference is, no surprise, the cases had a higher median PSA value pre-diagnosis, because they were on their way to cancer. Okay, so jumping right to it, this is what we call a Manhattan plot, a result from our Genome-Wide Association study. So the x-axis is basically chromosomal locations across the genome, right? So we've got each chromosome numbered 1 all the way up to the sex chromosomes there. And then the y-axis is minus log base 10 p-values. Right, so what we're looking for, they call this a Manhattan plot, is you're looking for skyscrapers. So those have the smallest p-values. And you'll notice something that's quite unique about this study. Well, first of all, there's the line here is what the genetics community has determined to define statistical significance, which is, as an epidemiologist, this drives us crazy. But this is what they have. It's 5 times 10 to the minus 8. So it's effectively correcting for a million independent tests. Right, so that's what that line across there is. And what's unique about this picture is there's a lot of things above the line. A lot of things. And that's due to, first of all, that this is a big population that we're studying. Second of all, it's a quantitative trait. And we had a lot of PSA measurements. Not only are there a lot of things above the line, we had to actually break the y-axis here and change it, because this peak, which is showing a lot of things as being genome-wide significant that are all kind of correlated with each other, is less than 10 to the minus 200. Right? And this is on chromosome 19. Anyone have an idea of what gene that might be? Or associated with PSA levels? Well, it's the gene that encodes the PSA protein. So, no surprise there. It's KLK3, which is basically that. So that's what we would expect to see. And it's actually a fairly interesting